All right, so we are now being recorded. So again, welcome to the sixth video teleconference for BIOL 105L YC. In this video teleconference, we are going to have several agenda. The first one is to look at the calendar and see where we are and recall on what we've done so far in terms of the exercises. Second, we are going to supplement what I have posted in eLearn on SDS page. And since it's too early to have a self-quiz, right, because that's usually what we do, I am going to have sort of a discussion with you instead on the concepts on SDS page. And lastly, I think the most important part of this video teleconference is I'm going to give you already your midterm project. Okay, so I will discuss it in this video teleconference. And later on, of course, I will be posting the project in eLearn. But this will be the first hand that you'll hear me discuss about the midterm project. All right, so we are now on the second week of February. Um, February the 12th is going to be a holiday, thus we're doing the video teleconference today. And you are supposed to be starting already watching the videos, reading your manual on exercise 6, SDS page, as an analysis of the proteins in the human blood. Right. So the week after this week will be the midterms week. We don't have a written midterm exam in the laboratory, so that's why later on I'll give you your midterm project, and you can already start working on it as a group. And the deadline for that will be on February 27, okay, at the last Saturday of February. So here we are so far. We've already reached exercise 6, more than halfway of all the exercises for this semester. And that's SDS page. And I'd like to speak with you. And if you have any questions, ask me with regards to the processes that are done in SDS page. But before that, something related to SDS page, which we don't have the liberty to include in our laboratory exercises because actually your laboratory exercises have been cut down to 10 or so exercises from, I think there were originally 18 exercises in cell mol. So here's self-fractionation and the processes involved. Okay, so when we say self-fractionation, the main process is not so different from centrifugation. Okay, so centrifugation is the main process to be done when you want to fractionate a cell. Okay, do you have any idea? I'm sorry, any idea on what fractionation of cells mean? Anyone? Self-fractionation. If you've studied chemistry, organic chemistry, I think you've heard of fractionation as well done in crude oil to produce gasoline and kerosene. Okay, the how do you call that fractionation? There's a fractionation process. So, what is fractionation? Anyone has an idea?
Separation of components, sir.
Yes. Okay. So, I will be talking an overview no, on the process on how we might be able to separate the components of cells. Um, in the SDS page exercise, this was done no, partly. There's a method there, a methodology that involves breaking the cell and releasing its contents. But I will not tell you which because you still need to study it and view the videos. All right. So here's what you need to do cell fractionation. You have your sample of cells. No, any cell will do, but it's easier to break animal cells, those that don't have cell walls. So the whole cells are placed in a suspension of buffer. Okay, so of course they need to be in a buffer to control the environment's acidity, the pH, and they need to be moist, of course. The first thing that you need to do is to break the cells. And in this illustration, you see here how there's sort of like a hammer, okay, or a like a mortar and a pestle, okay, the pestle that is actually used to break the cells here at the bottom of the tube. Okay, so when the cells are broken, the organelles are released from the cells, and they will be floating about randomly in the buffer, as shown here in the middle, the homogenate. Okay, and this homogenate, since we want to separate components, we need to process this. And it's important to remember that not all parts of the cell are of equal sizes. There are those that are heavy, like the nucleus, and then there are those that are very light, like the ribosomes. So that idea alone will already tell you how you might be able to separate the nucleus from the ribosomes, or the other way around. And the main process is called centrifugation. Okay, so we will be using a centrifuge to do this, just like in SDS page. So here in the illustration, you can see here a unit for centrifugation, and that's G, 20,000 G. That means 20,000 times the force of gravity, okay, on Earth's surface. So if that's the gravity, if that's the force that we are going to apply to the sample, it's gonna pull down the heavier components at the bottom of the tube, as you see here, and only the lighter components will be floating in the supernatant. Okay, so this portion that have stuck to the bottom of the tube, this is known as the pellet. Okay, that's the pellet. And the liquid that stays here on top is called the supernatant. So here we see the supernatant. So in this example, the pellets would include the nuclei, the heavier organelles, while the supernatant could include the ribosomes. And now you just use your micropipette, you aspirate all of the supernatant, and you leave the pellet behind, and voila, you already have separated the ribosomes from the nuclei. Okay, so that's the concept of cell fractionation. And then you can even separate even the smaller particles. Okay, but in order to do that, you need to centrifuge your sample at a greater speed and at a greater G. In this case, 50,000 G at 2 hours. Okay, 50,000 times the strength of the Earth's gravity, pulling all of those organelles down, so that even the smaller organelles will become the pellet, and only the smallest particles or molecules will stay in the supernatant. And then you can use extraction again using a micropipette. You remove all the supernatant, and you are able to separate the pellet from the supernatant. Okay, so the choice on what G's should I use, sir, when I want to separate organelles, it depends on what you are trying to get. For example, if you're trying to get nuclei, then do not use a very high G value, because that means the nuclei will be settling at the bottom. No? Or if you use a high G value, then you remove the supernatant, you only work with the pellet at the bottom, where you expect the nuclei will be. And for example, if you're trying to separate chloroplasts, okay, you might need to use a higher G, okay, than what you used for separating ribosomes, nuclei, for example. Okay, so um, that's the concept of self-fractionation. So you can be creative with it. It can be that your your organelle of interest will be in the supernatant, or it could be in the pellet. And that's when you decide which one to keep, the supernatant or the pellet. All right. If you viewed the videos in SDS page, that decision was also practiced. Okay, I think we had to centrifuge 2 times in SDS page, and you have to observe which one was removed, which one was kept. All right. So um, when you actually do self-fractionation, you don't simply use water as the solvent or the medium. You need to use something that is of known density. So in this case, this is sucrose solution because we know the density of sucrose. So you can see here at a certain speed, 65,000 Gs for 2 hours, the lysosomes, the organelles called lysosomes, would be near the top portion of the column, while the mitochondria will be in the middle, while the heavier peroxisomes will be near the bottom. Okay, so that's how you do self-fractionation. And of course, the centrifuge is a tool, and there's a reason why it's called the centrifuge, because it uses centrifugal force, the force that tries to pull away from the center, to settle down the heavier sediments, no, in your cell homogenate. So that's centrifugal force, the opposite of which is centripetal force. And these are actual microcentrifuge tubes. And you can see here the pellets that are stuck at the bottom of the tube. And here on top, we have the supernatants. So depending on what you're working with, you might want to keep the liquid on top, or you might want to keep the pellets at the bottom. All right. So are there any questions with self-fractionation and centrifugation? Any questions? Well, for blood, it's very easy to do. Okay, because we know blood is actually a colloid, meaning to say it's not liquid throughout. There are particles suspended in liquid in blood. It's like milk, it's colloid. Okay, so when we tried to do SDS page, okay, we actually were able to separate the components of blood, the solid cells, from the liquid serum or plasma by simply doing centrifugation. Okay, so when we analyzed for proteins in SDS page, we analyzed both portions of the blood, both the supernatant or the serum and the pellet, and we sort of made a cell lysate out of the pellet. Okay, so both of these contain protein. So for example, in the supernatant, you could have albumin there as a protein. And in the pellet, you can have here the proteins that are stuck to the cell membranes of the red blood cells or the white blood cells. So in SDS page, of course, the first part of the name of the process, SDS, is for sodium dodecyl sulfate, which is acting like a detergent. Okay? The sodium dodecyl sulfate both has a hydrophobic tail and a hydrophilic head, which when mixed with membrane proteins, the hydrophobic tail will get stuck with the proteins. And when you wash it away using an aqueous solution, it's gonna get washed away because the hydrophilic heads are gonna go with the hydrophilic solution. Now, once SDS is applied onto your protein samples, it's gonna stick on your protein samples and incorporate therein negative charges. Okay? So your protein sample will sort of gain a negative charge when they are mixed with SDS. Not only that, they are also going to unfold. Okay? So notice here how these proteins, A, B, and C, have unfolded when SDS is added to them. Okay. So once the proteins have been unfolded, you may then start analyzing them, no? And that's where the PAG portion of SDS page comes in. What's the meaning of PAG?
Polyacrylamide gel, sir.
Al- Correct. So the polyacrylamide gel. If you view the video, it's already actually a pre-packed gel, meaning to say it comes in sachets that you just open. Okay, but we can make it ourselves, but it's difficult, so we just buy the pre-packed gel. Okay, later on there's another activity called agarose gel electrophoresis. We're gonna make our own gel. I will show you that in the video. So here the polyacrylamide gel has wells, as you can see here. So it looks like the top of a castle. Okay, and in these wells we put our sample of proteins. Okay, so the proteins are already negatively charged. Now the gel, you put it in the electrophoresis chamber, and electrodes are going to be attached on the gel. On the top you have the negative electrode, and at the bottom the positive electrode. What's another name for the negative electrode? Anyone knows? Starts with letter C.
Cathode, sir.
All right, the cathode is positioned near the wells, while the positive electrode starts with letter A.
Anode, sir.
The anode is positioned at the bottommost part of the gel. So when we turn on our electrophoresis apparatus with the tank buffer, and we have the protein samples here which are negatively charged, they are going to be attracted towards our anode. So they're going to move down the gel. But their rate of movement depends on their size. The lighter proteins or the smaller proteins are going to move further down the gel than the heavier proteins. Okay? And that's how we judge which proteins are present and what their sizes are. Okay? In our SDS page. All right. So the gel is actually able to do that, because if you look at here in the illustration in letter B, the gel is a polymer. Okay? Meaning to say it's not a solid substance. There are spaces in between the gel molecules that allow the protein molecules to percolate, right? That's the term, if you're familiar with the term percolate. It's gonna go through the spaces in between the polymer of the gel. Okay, so we put the sample here on top, and then when we turn on the electrophoresis apparatus, the lighter samples will have an easier time to go through this mesh of polymer, and then the heavier samples, the bigger samples, will have a difficult time going through and will therefore stay near the wells after the electrophoresis. Now, the sizes of the proteins are measured in terms of daltons, okay? Now, because they're so big for molecules, we measure them in kilodaltons. Okay. And it says here that the size suggests the identity of the protein. Okay. So you just have to trust that proteins of different types have different sizes. So that's the limitation. You cannot really identify the protein unless there's already a standard that you use in gel electrophoresis. Thus, we have a standard, but our standard is sort of limited. It only tells us the sizes, but not really the identity of the protein. Okay? And the standard that we use has a brand name. The brand name is Precision Plus Protein Kaleidoscope. Okay, so that's the standard that we are going to be using. If you've already reached the videos, the end of the videos that I posted, you'll notice that our end product unfortunately doesn't look as appealing as this one. Who among you have seen the picture already of our finished gel? Nakakita na kayo? Someone was sending a message. I heard the messaging sound. Wala. Okay, again, back to my question. Have you seen the result of our SDS page?
Yes, sir.
All right. So unfortunately, it wasn't done perfectly by myself, or maybe I did that in purpose. So it looks really blotty and not so clear. But I provided you the picture so that at least you can estimate the bands, where the bands are. And on the first well would be, sorry, on the first well would be the standards, not the sizes. Um, a perfectly electrophorest sample would sort of look like this, no? Where the bands are completely separate from each other. Okay, so I leave the analysis to you, no, as to how the proteins from the lysate are different from the proteins from the serum of the blood based on our result. So just a review, we use 200 volts, no, that's our voltage, at 400 amperes for 40 minutes. So we ran it for 40 minutes. And our staining after running the gel electrophoresis, we remove the gel, we place it in a trough, we place a stain. The stain is again Kumase Brilliant Blue. No, the one that sticks to the protein R chains. Excuse me. And then we destain the gel. Now we remove the stain and then we were able to view the final result. The one that is at the end of the last video that I posted. So here are some deadlines. For the SDS page exercise, the deadline will be on February 13. Maybe this is reasonable, I think. Okay, February 13. It's not that difficult. The midterm project, which I'm gonna reveal to you now after this, will be due at the end of February, February 27. Okay, so before I reveal to you the project, are there any questions on SDS page? Anything you'd want to clarify from the videos that I posted? Questions or concerns?
Excuse me, sir.
Yes.
Since you said man nga ano, dili appealing tong naa sa imong video presentation. So asa na lang mi mag-base sa mong kuan, results or analysis?
All right. Thank you for the question. Yeah, it's not appealing, but there are still bands visible. Okay, so you base your analysis from those bands. Okay, and you may include in your analysis the reasons why it turned out that way. Okay, so I've shown naman the methods to you. I've shown to you how I loaded the samples, how there were some contamination. You've seen that. So you can include that in your discussion on the probable reasons as to the result. So do not do not just download a perfectly formed electrophoresis gel from the internet. You base your analysis from the actual one, the not so appealing one that I posted, the actual one. Is there a follow-up question to that?
Na. Thank you, sir.
Okay. So actually, it's not a guarantee that I'm the one doing it, that it's beautiful. Last school year, of course, the students, the third years now, we're the ones who actually did this themselves, no? And their output is actually quite more pleasing to the eye than the one that I had. Okay, so, but I believe given another trial, another chance, I can perform, I can do it better. Any other concerns on the SDS page? Okay, if there are none, let's proceed to your midterm project. Okay, so this one is due on February 27, 2021. Okay, so in this project, in the prelim project, you designed your own methods. No, so the nature of this midterm project is a bit different. You will not design your own methods anymore. Instead, you will be looking for a published research paper that uses the methods that I'm asking you to look for in the research paper. Okay. So you're going to be reviewing a research paper or two research papers, no? Um, in the field of biology, of course, no? Because you could review a paper from chemistry and the methods are very similar. There's still centrifugation. So don't do that. The paper should be in the field of biology. Um, better if cell and molecular biology in a field, but it can also be in microbiology and the other related fields. So you get a paper, you can download papers now from ResearchGate. I don't know if you've been to ResearchGate. Many papers there are for free. You can just get a copy um, and try to read through the paper and see if what I'm asking you to look for is present in the paper. Okay? Now, if that methodology is present in the paper, you can then choose that paper as your paper to be reviewed. What do you need to focus on? Number one, the objectives of the research. Now, it might take some time for you to find what the objective is because usually the research papers that are published, they don't outline the objective like how you would do in your thesis paper, di ba? They won't say the objectives of this study are A, B, C, D. They won't do that. It's all in a paragraph. So you might need to read the whole paper first before finding out which ones are the objectives. So you need to know the objectives. Number two, the methods that are used to answer the objectives. Number three, the results. And lastly, you need to look at the conclusion that the author wrote with regards to the objective based on the results. Okay, so what am I talking about here? So let's specify this. Um, but before that, when you submit your output, you include a complete copy of the paper that you're reviewing. Okay, so that's how you'll submit your output. So what am I asking you to look for in the paper? Okay, the published research article or articles should include in its methodology the following. Letter A, spectrophotometry. Diba? We've done that in spectrophotometry of proteins. Take note, you are not, if the paper is using spectrophotometry for another reason aside from Bradford assay, that's fine. Okay? It doesn't have to be limited to Bradford or protein assays. No, so it can be any other application in biology as long as there's spectrophotometry. All right. Letter B, SDS page. No, na method. Again, not limited to human blood protein analysis. It can be SDS page and then they're using a different sample of proteins. That's possible. So, I said here research article or articles because if both of these methodologies, spectrophotometry and SDS page, are found in one paper, you can already settle with that one research paper. But if one paper only gives you a methodology on spectrophotometry, then you need to look for another paper that uses SDS page in its methodology. Okay, so one or two papers. So in your review paper, include the following: an overview of what the research article is about, the problem that it was trying to address. Now, in your own words, so please don't copy paste the words from the research paper. I want to read the words as if you yourselves wrote it, or that you really wrote yourselves, okay? Not copy pasted from the research paper. The specific objectives that is addressed through the use of spectrophotometry and or SDS page. So for example, the research paper could have 10 objectives. You don't have to copy all 10. Just focus on that objective that was answered using either SDS page or spectrophotometry. So, meaning to say, you are not really reviewing the whole paper, guys. You are just reviewing the specific method, the objective that is answered by the method, and the results and the conclusion based on that method. Okay. Um, Thirdly, a schematic diagram of the actual steps done in the research. So schematic, so meaning to say still similar to what you're doing in your laboratory reports, schematic diagrams. Then, the specific name of the protocol used, if any. So for example, di ba, Bradford assay, that's a specific name of a protocol. But if there are no specific names given in the research, that's fine. Okay. Then you point out the specific results and how the researchers presented and analyzed and interpreted the results. So you also say that in your review. And lastly, the conclusion that are addressed, okay, or that addressed, the conclusion that addressed the specific objectives that you focused on a while ago, when you focus on the objectives. So these are the things that you need to consider for your midterm project. Are there any concerns or questions about this one? As to how current the paper is, I'm not going to be very strict with that because the main focus of this exercise is for you to see how the things that we do in the laboratory are applied in real researches. No? So it's okay na yung paper siguro mga early 2000s. I think that will be fine, no? Because you are just reviewing the paper. You are not making a new topic. I'm sure you have questions and concerns about this one or clarifications. I'll give you time to think. If there's none, anyone from the group can just say there's none.
Excuse me, sir.
Yes.
I actually dili ni siya question regarding sa research, but is it okay nga mag-contact lang sa imo in case lang nga kaning naay questions outside aning synchronous meet?
Yeah, sure. Okay, yeah.
Sir,
I would
sir Gmail lang, sir.
e-learn. Pwede
Okay.
e-learn, no? Pwede rin Gmail. Pero I think mas faster pag e-learn.
Okay, sir. Thank you.
So that is expected. Of course, right now, your brains are still thinking, you're still looking at the text, and you cannot formulate a question or concern. I expect that. So again, correct yon. You may contact me anytime through eLearn or through Messenger. I also reply through Messenger and email about any concerns about this or your exercise number 6. But right now, wala na talagang question para live ko, I can answer it live. I will be posting this in eLearn. So you will also have your own copy. Plus, this is recorded so you can view this video teleconference again later on because I will post it in eLearn. Do you think this is doable, guys? Ako na lang ang ask.
Yes, sir.
All right. Okay, so just remember our goal here is for you to see how the things that we are doing are applied in real researches, no? That's really the goal here. All right, sige. So, um, again, if you have any other questions, you just send me a message. And for now, I think I'll leave the thought to cook in your head. Okay? So you might still be thinking, contact your group mates, talk about this already. The deadline is at the end of February. You have ample time to do that. There's no specific um, there's no specific format again. Okay? Except for the schematic diagram, you know that I require it to be simple but thorough. Okay. And of course, past tense because you are reviewing something that was already done. So past tense. Okay. So I thank you very much, guys, for joining this video teleconference, even at this early day of this week. And on Friday, we won't be meeting in a video teleconference, so you can rest um, and have a productive day ahead. As soon as we end this, maybe you can talk with your group members already. So, guys, thank you for joining, and you are now all dismissed.
Thank you, sir.
Thank
Take
you,
care,
sir.
sir. God bless.
Sir.
Thank you, sir.